Hi, I'm Dan Zlotoff. I'm one of the cardio-oncologists at Massachusetts General Hospital and part of the Atrium team. I'm going to be describing the collection of the blood research samples at this point. Patients enrolled in the trial will have multiple different types of labs being collected. Some of these are standard of care labs collected by the healthcare team. Others will be the safety labs, which are prescribed by the protocol. And third are the research sample labs. I do want to make clear that while we certainly encourage all the patients in the trial to have research sample labs collected, it's not absolutely required. If, for example, a patient were to come in the middle of the night and be randomized at that point, and the resources are not available to collect research labs, that's okay. We certainly would much rather have the patient be enrolled in the trial, even if research labs for certain dates can't be collected. The Atrium trial offers a unique opportunity to collect biological samples from patients with immune checkpoint inhibitor myocarditis. These samples we expect would yield tremendous insights into the disease pathogenesis as well as useful biomarkers. Our plan is to collect both blood samples and histologic sections. From the blood will be segmented into plasma, serum, and peripheral blood mononuclear cells. And the tissue will be derived from both tumor and myocardial biopsies when those are performed and available as part of routine standard of care. So starting first with blood samples. This slide shows an overview and the schedule of the research samples we intend to collect for each subject. For the first of these will be on visit 1, which is day 0 on the day of randomization. And there are 4 different research samples that I'll be covering. On visit 1, all 4 of these research samples will be collected. Visit 2, which is day 1, no research samples are collected. Visit 3, which is day 5, research samples 1, 2, and 3 are collected. Visit 4, which is day 14, research samples only 1 and 2 are collected. And on visit 5, research samples 1, 2, 3, and 4 are collected. As you can see, there's a total of 13 research samples that are collected over the course of the study. And each research sample will be divided into 3 aliquots. Hence, there are 13 samples with 3 aliquots, so 39 total aliquots to be collected by the research site. To just show this a little more clearly, this is the schedule of collections. And there are the 4 research samples are shown in the grid at the bottom. Research sample 1 is a plasma tube, which is a K2 EDTA tube, purple top. Research sample 2 is a gold top serum separator tube. Research sample 3 is has a black and blue top. This is called a CPT tube to collect peripheral blood mononuclear cells. And research sample 4 is another EDTA tube or purple top to collect plasma. Focusing on day 1 again, research samples, all of research samples 1, 2, 3, and 4 will be collected. On visit day number 2, there are no collections. Visit 3, research samples 1, 2, and 3 are collected. Visit 4, research samples 1 and 2 are collected. And finally, on visit 5, all research samples 1, 2, 3, and 4 are collected. I'm now going to go through the list of supplies that the Atrium sponsor will be providing to each site as part of the research sample collection process. So first, EDTA 10 milliliter tubes for collection of plasma. Then gold top 5 milliliter serum separator tubes. Then 8 milliliter CPT tubes for the collection of peripheral blood mononuclear cells, 5 milliliter pipettes, and disposable 5 milliliter transfer pipettes. And I'll add that sites can choose to use one of these or the other for liquid handling. For those sites that don't have pipette controllers or familiarity with those tools, the use of the transfer pipettes is totally acceptable. Cryovials for the storage of the samples and shipping back to the Atrium biorepository. 15 milliliter plastic conical tubes, an 18 gauge needle, and 5 milliliter syringe. These are for drawing the CryoStor cell preservative solution as part of research sample 3. A cool cell freezing container. This is for a controlled freezing process of the cells so they don't get damaged. Additionally, we'll ship a plastic storage box for shipping the cryovials back to the Atrium biorepository. Additional supplies that I won't list by name include supplies for blood collection from the subjects, as well as shipping supplies. Additionally, I do want to mention we will be sending a lab manual that will provide all the details about the lab collections, as well as there are additional videos available that demonstrate the lab collection and processing steps. Part of the Atrium study, we are asking the sites to send us tissue specimens from both the primary tumor that the subjects have, as well as myocardial specimens when biopsies were performed as part of routine clinical care. So we ask that these histologic sections be sent in slide boxes that we will provide to the sites. We will be providing 2 slide boxes, one for the tumor sections and one for the myocardial sections. Additionally, we will provide pre-labeled FedEx bubble padded shipping envelopes for these boxes to be sent back to our repository, as well as the histologic slide inventory form, and finally the instructions for how to package and ship these slides. These slide kits will be sent to the sites upon patient or subject randomization, not ahead of time before or as the sites are being set up. The collection process will be that each site will request slide-mounted histologic sections from their pathology departments upon patient randomization. Standard fixation methods to preserve the nucleic acid integrity is required, and typically this is 10% neutral buffered formalin for 6 to 72 hours. For the tumor sections, we ask that sites send 10 unstained slides. These are positively charged and unbaked at 5 microns thick, and also 1 slide that is stained with H&E. If sites are unable to send 10 unstained slides, then we ask that they send as many as are available. Slides should be reviewed by a licensed pathologist, and ideally there is more than 50% tumor fraction present on these slides. For the myocardial slides, we ask that sites send 10 unstained slides, both positively charged and unbaked at 5 microns, and 1 slide that is stained for H&E. Again, if the sites are unable to send 10 unstained slides, that's fine. Send as many as are available. For storage of these slides, each kit will again contain 2 slide boxes. One will be labeled when it arrives to the sites for myocardial sections, and one will be labeled for tumor sections. The boxes are specifically designated for the enrolled subjects, so we ask that they not be used for any other study subjects. Site staff will provide these boxes to their pathology department colleagues and request that the designated slides be placed in the appropriate box. To be clear, individual slides do not need to be labeled as long as they go in their designated box. Following the return of the slide boxes with the requested slides and sections to the site PI, then they can be shipped back to the Atrium repository in the shipping supplies that are provided.